very good afternoon. Welcome again to yet another talk under JIGYASA-ATL webinar series. In this talk, as all are aware, we are having very interesting lecture by eminent speakers. You get an opportunity to interact live with the scientists working in different domains of science and get answer to queries. In this series, today we have with us Dr. Sucheta from CSIR IICB, which is one of CSIR laboratory in Kolkata. So let me first take this opportunity to introduce Dr. Sucheta Tripathi. Dr. Sucheta is a computational genomist currently engaged in deciphering genomes of thermophiles grown in India that are known to harbor a plethora of commercially important enzymes. Her team has sequenced and analyzed 20 species of cyanobacteria and fungi, highest number of genomes from a single lab from India. Her group has created many resources for hosting and analyzing those genomes. The software package Genome Annotator Lite, also known as GAL, is created as containerized package for the ease of installation in any system in a system independent. Currently, Dr. Sucheta is working at CSIR IICB as a scientist. Prior to this, she worked as the lead genomic scientist for oomycetes pathogen genome at the Virginia Bioinformatics Institute at the Virginia Tech for a decade. She has been awarded the prestigious Ramalingam Swami Fellowship for returning back to India. In India, 5 PhD students have already graduated with her. 2 postdoctoral scientists trained in her lab have got permanent position in India and abroad. In her lab, 22 master students have been trained. She has altogether published 50 research papers and 4 book chapters. Her work has been cited 3168 times already. She is now serving as a senior editor at Molecular Plant Microbial Interaction and also as an associate editor of Frontier in Genetics and Frontier in Microbiology. She is also serving as the research advisory team member of National Tea Research Foundation. So today she is going to speak on bioremediation with indigenous microbes. So first of all, I request you all to kindly keep yourself muted and your video off. And if you have any question related to today's topic, that question in YouTube or Webex chat box. I will take this question with the speaker after her talk. With this, now I invite Dr. Sucheta for her talk. Over to you, Dr. Sucheta.
Thank you very much, Dr. Pooja and Dr. Khalko here and the JIGYASA team for inviting me here for presentation. I'm really excited to share some of the work that we are doing here in IICB, and our work is mostly, as she said already, that we are interested in extremophiles and deciphering the genomes of extremophiles, but we also do work on the power of the bioremediation. That is one of the biggest problems that our country is facing right now, using these indigenous microbes. And if we are talking about indigenous microbes in India, we know that we have actually known nothing about the existing species. And India is a large country with many numbers of species, which can range between 7 to 17% of the entire species of the entire universe. So this is called as a mega diverse nation. And with this, I would like to just walk you through what nature can do for us. And before I get there, I just want to impress upon you about the origin of species on Earth. If you look at the history of our planet Earth, you will see that it is about slightly more than 4.5 billion years of age. And then after that, many events have happened. And that time when the Earth was formed, it was just a rocky planet. It didn't have anything. It didn't even have water. And there are many conflicting hypotheses that tells that how water came into the our planet, and some of that says that some comets hit the Earth and they carried the water, and that's how the water began to flow in our planet. So after the water started coming, and the region in which our planet exists, it is called as a Goldilocks zones. So this is very favorable for creating lives. And exactly how many years back the exact life was created was not known, but the estimated time is about 3.8 billion years back. And earlier people believed that the earliest form of life was RNA. But now there are conflicting hypotheses. Some say proteins, some say RNA. So from there, it started, self-replicating molecules started coming, and then smaller forms of microbes started coming into this planet. And then the next thing happened was there was a methane buildup in our planet, and that was being uh, since that was being utilized by some smaller form of smaller life forms, that could be a bacteria or could be an archaea, which happened slightly after the earliest form of life came. And then the viruses came, and slowly, slowly, as you see that, then the great oxygenic event happened on this planet Earth, where the photosynthetic organisms, the small photosynthetic organisms, the cyanobacteria, they came into existence, on which actually our lab is focusing on. And when the carbon dioxide was started fixing, then the oxygen concentration of this planet started increasing. And oxygen, as you know, decreases the atmospheric temperature, and the whole Earth came into a big ice age. And then again, everything started to fall apart, and life form started decreasing. And then slowly, slowly, the ice melted, and then smaller organisms started coming. About some years back, then after some time, the sulfur reducing bacteria came, and after that the multicellular organisms started coming, then the alga, and then the land plants. And if you see the history of human life, it is not beyond 600 million years back. So about 600 million years back, 6 million years back, the humans started coming into this planet. And the modern humans came into existence about 50,000 years back, when we started speaking and we started communicating and we started analyzing. So this is a brief history of humankind. And if we are talking about science, the science is not even few hundred years old. And with this knowledge, we cannot claim that we know everything. Since the nature has been doing this for billions of years, and nature is doing it in a different way. Nature is doing by hit and trial method, in which it is just creating some life forms, and if it is finding the life form is not suitable, it is just eliminating. And that is called as a natural selection. And natural selection is a process in which we have the most successful species existing right now, and the ones that was not very successful were eliminated. Something similar to dinosaur and other species. So why I'm talking about all of these things? The reason is, when we are talking about science, or we know something we want to develop, which is like for the betterment of the humanity, we are just relying on science. But if you look at the nature, which has been doing this experiment for more than billions of years, so that is having much more knowledge than we can even think of. So taking a cue from nature, many things scientists are building these days, and they are smart enough to think that nature knows better than us. For example, if you look at here, the left side, this is a picture of a lungs. And the lungs, the function of lungs is diffusion of gases. So all it does is when you are inhaling gas, it goes into the alveoli, and then it diffuses gas into the blood and exchanges that. And in doing that, it wants to do it in such a way that it is most efficient. That is, it is reaching out to every part of the lungs. So if you look at the design is something like this. And these are called as the acini. And taking a cue from here, scientists have built capacitors and nanomaterials which can reach out to different parts, or to increase the surface area. They have designed semiconductors and other electronic devices from this experiment. So as you can see that this is a very, very nice way of imitating nature. Similarly, in building structures or buildings, nature's cue has been taken. For example, the first one is the Lotus Temple, that is in India, and the second one is the Gharkin in London, which is inspired by the structure of a cucumber. And the third one is the Beijing National Stadium, which is inspired by the bird's nest. And bird's nests are very resilient, during storm and many things, it just continued to stay. It's just hangs on. So they have taken a cue from there here. And Olympic Pavilion of Barcelona was also designed from nature, taking its shape of its fish. And this one is very interesting. This one is called as the East Gate Development in Zimbabwe. What they have done is they have taken a cue from the ants mound. Ants mound is structure. If somebody has to see it, you'll find that amazing engineering there. Engineering is there. How it happens is, it's a structure. It's a three-dimensional structure, and they have engineered it in such a way that the internal part of this mound also get equal amount of oxygen. Uh, Temperature is controlled very well inside this. So using this, they have built this building in which they are not using air conditioning. So these are some of the examples. The reason I am saying all about this is we are also trying to imitate nature in not building structures or something like that. All we are trying to do is we are trying to take the organisms that are growing here as extremophiles, and we are using a technique called as reverse genetics. What is reverse genetics? Genetics is a science when you are studying the genes, and then we are going for understand. You know the characters first, what the genes are doing, and then you study genes, which is called as forward genetics. Reverse genetics is something where you are going directly into the DNA, which is the genetic material, and we are trying to sequence that. So nowadays, the sequencing technologies are very, very well advanced, and it's very easy for labs like us to sequence genomes and analyze those quite easily and decipher it. So all we are doing is we are trying to gather all these organisms which are growing here and there, and we are seeing their characteristic, and we are trying to understand first what is there in their genome, and then we are identifying the genes, and from the genes we are trying to go to the function. And if we find some function is very, very useful for us, then we try to utilize it for betterment of humanity. So I'll just share you a story of two fungi and one bacteria. So here, this is a story started when I joined IICB about 7.5 years back. I had a senior colleague, Dr. Suman Paula, and she was interested in fungal biotechnology, in which we are working with a fungus called as Torbidomyces. And these organisms are very, very interesting because these organisms grow in ants mound, and they have a mutualistic relationship with the ant. And as we all know that the fungi, they have a unique ability to degrade cell wall. Cell wall is the biomass that we get in abundance in India, but mostly what happens, people burn this biomass to create, the stubbles are burned. That is why we get pollution and we get a lot of problems. But these stubbles can also be used for production of biofuel. But why we are not doing it? The reason is the stubbles are mostly made up of a material called as cellulose. And this cellulose, if you want to degrade into smaller components, needs more energy. So by way of degrading cellulose and producing ethanol, the amount of energy you will spend is actually much more than the energy you will derive out of the ethanol. That is why people are interested in actually getting the enzymes that are produced in fungi. And this is a huge area of research, and obviously Dr. Kolas was very interested in working in this area. And then I wanted to collaborate to study the genomes of these organisms. And when I collaborated, they had this lab culture which was looking something similar. And it looked like Dermatomyces mycelium. Mycelium are the structures that grows underneath, and then it becomes a fruiting body, what you call it as a mushroom here. So we took it and we analyzed them under microscope, and we found that it looks like this. But actually, Dermatomyces, which belongs to a class of fungi called as Basidiomycetes, should have something like a clamp connection. That is, a clamp-like connection between the mycelia, which we did not see here. And that raised an alarm, and we thought whether this is actually Dermatomyces or something else is happening. So immediately we took the cue and we wanted to go back and check the genomes of this. So we extracted DNA. Then we extracted DNA. Uh, there is a region of the genome which is quite conserved called as the ITS region, and we amplified that using a primer and we sequenced it. After sequencing, there is a method that is used largely, and now there is a server called a GenBank server which is hosted at the NIH, National Institute of Health in US. So what we do is we take this sequence and we run a program called as BLAST program. BLAST does a similarity search. So your sequence will be searched against the existing database, and that will tell you how much similar it is to the existing data set. And from this data, all we came up is this organism is very close to something called as Artemia malaysianum. Malaysian, Malaysian. So this organism is different from the Termitomyces that we wanted to work on, and this belongs to a different class of organism. So our interest, our journey began with this organism, and we started to do several other things, including uh, we started we started analyzing this organism, and we have done several other studies that I'm not presenting here because the topic is to study about bioremediation. Dr. Rajiv Majumdar, who was a graduate student in our institute under Dr. Suman Kawla, he was working in different aspects. So he joined my lab with the CSI fellowship, and he took up in a very different direction this particular work. What he did, he took the biomass of this Artemia malaysianum, and then he started to create this dried biomass, which if you grow for a many days, you get a very little biomass. So he started collecting this dried biomass and he started doing experiments with that. And accidentally he found that when he is treating this biomass on chromium, and chromium, it's a pollutant. It is coming out in the leaching process of leather and many other tanning industry, many other industries. They produce this as a heavy metal and it causes a lot of problems later on when it is mixing with drinking water. So after this, when he treated the dried biomass with chromium solution, he found that chromium is adsorbed to the cell wall of this particular. And we are very excited with this. And then he started doing a lot of other experiments. I am not getting into details about that to understand actually what is happening, whether this chromium 6, and he also found that this chromium 6 is reduced to a less toxic form, that is chromium 3, using our biomass. Then he wanted to know exactly what is happening, whether the cell wall is acting upon it or whether it is a physical phenomenon or it is a chemical phenomenon or whatever is happening inside. And then it was found that this chromium 6 species is actually reduced into chromium 3 on the cell wall, and it is a surface phenomenon. And there are three independent variables. One is a contact time, which is A, and the biomass and the pH. These three variables determine how much of biomass will be needed to absorb maximum quantity of chromium. So he came up with the equilibrium, and this was published in scientific papers. This is very well cited paper, and this is very well taken, and this was also this came in Hindi. So with that, and this is a picture to show you exactly some images from the SEM analysis to show this is the surface phenomenon, and then he had done other experiments like XRD, FTIR, Raman spectroscopy, and etc. to understand what are the other molecules present on the surface and what exactly is happening here. And with this, we are very excited. We wanted to check what happens, whether this organism has any potential to absorb other heavy metals, because chromium is not the only heavy metals that is occurring in India. So we went ahead and checked with nickel, iron, copper, cobalt, lead, and arsenic, and to our amazement, we found that it can actually absorb all other heavy metals and more so to the lead. And this work is under progress. We want to see that whether we can develop this into a biomaterial, because we cannot go to the tannin industry and just spill the powders. That's not the actual good way of doing this for bioremediation. So we have collaborated with
Dr. Sucheta, I kindly request you to come close to the mic because when you are going away, your voice is not coming clear.
Okay.
Yeah,
Is
now
it
it's
okay?
very clear. Yes, yes.
Okay. Thanks for telling. So, with this, we found that this organism is actually having a great potential to act as a bioadsorbent to many heavy metals, and we collaborated with CSR NML, and we developed something called as bio beads, which is embedding some synthetic material into the biomass, and this is very lightweight. In fact, Rajiv tried with many other methods. Rajiv created alginate beads and all those things, but we did not find any promise. So we went for material development, and we have come up with these beads, which can be really useful for treating this. And we have tested their properties, and we found that they are maintaining the abilities to absorb all the heavy metals that we are interested in. So this is the picture to show what happens. So now I'll move to the next story. That this story is not entirely homegrown in our lab, but it is a collaborative effort with another lab in Calcutta University. And these are the three amazing ladies who contributed mostly to this work. And here, this is Dr. Anindita Sil. She is a professor in the Calcutta University Biotechnology Department. And this is Dr. Dia Sen, who was my postdoc. Now she's working in Sion, New Zealand. And this is Cornelia Paul, who is a graduate student of Dr. Anindita Sil. So the story begins like this. Anindita went to some uranium mine, and she found that this typha, this grass is called as typha. You would have seen this grass growing everywhere with white flowers. So not white flowers, I think brown flowers. The inflorescence is brown. So this grass is very much wild, and it grows, and it also causes bioremediation. It cures the land of many other problems like such as heavy metals. So Anindita found that this grass is growing despite presence of uranium in this area, and she took the sample and she brought it to the lab, and she found that this grass is containing a fungus, a yeast. And then she was very much interested to study what the grass is actually doing with the yeast is doing with the grass. So that yeast was an endophyte. What is an endophyte? You would have seen fungi, right? The fungi can be of three different types. Fungi do not have photosynthetic apparatus, so they need outside sources for food. Either they can live in other organism and establish a mutualistic relationship, that is, they benefit from each other, and endophytism is such one such way. And another one is saprophytism, where fungi lives on dead matter or dead mass. It doesn't kill the organism, but it lives on already dead organisms, and then it dissolves it and it consumes it as a food. And the third way is parasitism, where it lives on the living organism. And when it is living on the living organism, it uses the host's machinery to produce its own components that it is interested in growing. So this organism that she had discovered is a yeast, and it is named as Rhodotorula, and it is a endophyte, that is growing inside the plant, and also the plant benefits out of this organism, having a mutualistic relationship. And why it is bioremediating? Because it is, first of all, it is growing in the uranium mines, so it must be doing something for the organism so that uranium is degraded and the land is becoming more and more usable. And another way could be it also has the ability to dissolve inorganic phosphate and other inorganic forms. For example, if you go to the lands where you will find some of the mineral deposits which is not solubilized, and that land mass becomes totally unusable. And presence of such organisms which can actually dissolve the undissolvable or inorganic matter, which can also help in making the land rehabitable. Here is an experiment to show that this is a media in which phosphate is existing in a insoluble form. but you see the halo all around this organism. That means that this organism is actually solubilizing the phosphate which is present in this media. And this is a picture to show, although this organism grew in typha, which is a grass, which is a monocot, when they inoculated to rice plants, you can see that the rice plants had a very good growth enhancing effects, like the root growth and the shoot growth were very high, and it also had better yield. So that was inciting enough interest for us to study this further. And our lab, being the genomics lab, we were interested to study the genome sequences of this organism. And as I said, that there are many sequencing technology, and some of them are second generation and third generation sequencing, where the sequencing is done very, very quickly and easily. And it is just affordable by any small lab these days. So there are two different sequencing method we have used for this. One is a second generation sequencing method, producing this many reads. That is, the reads are nothing but when you isolate the DNA, you cannot sequence the entire DNA because DNA can be very, very long. So all happens there is the DNA is chopped off into randomly short pieces, and they are size selected, and those sizes were picked up by a chip, and in that chip each of the sequencing strand is multiplexed, and they were sequenced. So you can get a huge number of reads or huge number of DNA sequences from a small fragment. And the third generation sequencers are, there are several methods, and one is Oxford nanopore. Here what happens is, instead of chopping the sequences, all we do is we pass the DNA through a small pore called as a nanopore. And as the DNA passes, DNA has 4 bases, and each of the bases have a different charge potential. So as it passes through the pores, it generates electrical signal, and that electrical signal is recorded, and that determines the sequence of the genome. And this technology produces larger reads, whereas the second generation sequencing produces very short reads like 250 base pair. And then after getting this, the important or the most challenging step is to join them together. This is like a puzzle. You have to join these short pieces into a longer fragment, and this is called a genome assembly. And we have got a near perfect genome assembly for this organism, which is almost like 98% of the genome that we have got captured the whole genome. And then we did an analysis called as Bosco analysis, and this is a method in which we are checking whether the conserved genes are present, all of the conserved genes are present in the genome or not. If 100% of the conserved genes are present, that means the genome is complete. And if we are getting some values like 97 to 98 or 99, that's also very, very good score, as you can see here. So that means that we have captured most of the genome. So with this knowledge, we went ahead and analyzed the genomes with some of the already existing Rhodotorula species. And once a species was isolated from Antarctica. Antarctica, as you know, is very, very cold all the time. And then you can see the alignment results. It is actually 100 near identical alignment sequences are there. So this tells us the diversity of these organisms, and also this tells us the origin of this species, how this species came all the way up here. Um, and then we did find out that this contains the mitochondrial genome, and we compared that they have very, very good matches. So this tells us that this species was probably propagated by some travelers or something. And the very interesting observation we have got here is, as I showed you in my earlier slides, that this organism has growth enhancing effect on higher plants. So how it is happening? And nitrogen metabolism is one of the most significant metabolism on higher plants, and higher plants cannot fix nitrogen. And this organism, Rhodotorula, contains a transcriptional regulator, which is a switch which can regulate between two different nitrogen metabolic sources. For example, if the higher plant gets the nitrogen, external nitrogen, then a particular mechanism will be switched on, which will carry out a particular pathway to metabolize it. And when there is no nitrogen, this organism, Rhodotorula, will switch it on to another pathway in which it can start fixing the nitrogen for this organism and then make it grow. So that is why it is having a switch, transcriptional switch, and which is a very very essential gene for any plant, higher plant to grow. And this is what we have discovered through our genomic studies. And as I said, that Rhodotorula is an endophyte. What is an endophyte? Means that it enters into the host, it stays there inside the host, but it doesn't cause any harm to it. It rather helps it. So, but then in order to enter into any living organism, the first hand reaction happens is the host will react. The host will not let any pathogen or anybody enter into its body. It has a mechanism, inbuilt mechanism. So that mechanism is actually switched off because this endophyte has to enter into the host system. And in order to do that, they have developed is they have actually expanded a particular gene family which is called as proteophosphoglycans, and this family of proteins actually helps them to invade into different types of cells. So that is why you could see that they were growing in typha. They can also grow in rice, and they can also have a broader host range. And when we compare this with a pathogen, maybe you would have heard about Ostilagomedis. It's another fungi which is a pathogen which destroys wheat crop, and this pathogen causes damage to the host. Although it enters into the plant, but it doesn't help the plant; it destroys it. It's a parasite. So when we compared this with the Ostilagomedis, we found that there is a destroying part in this genome which is called as a gene cluster, which is called as 19A. This entire gene cluster is deleted in Rhodotorula. That means it has adapted itself in such a way that it enters into the host, but it does not cause any harm. And apart from that, it also has many many mechanisms, like say when there is a pathogen or when there is another organism is trying to enter into the host. Say for example, in case of plants are very sensitive to fungi, and the fungal cell wall contains an element called as chitin. And if the plant sees that there is a chitin nearby, then it releases an enzyme to degrade it. Chitin deacetylase degrades it. And Rhodotorula, very cleverly, it has designed its system in such a way that its chitin is converted into chitolase. So once chitolase is there, plant is unable to recognize it. And since plant cannot recognize it, this can easily enter into the host. So this is surveillance mechanism this system has developed. And also plant's first hand defense mechanism is something called as salicylic pathway. So it degrades the pathogen. But then Rhodotorula has evolved a mechanism in which it degrades salicylic acid by producing an enzyme called as salicylic hydrolase. Thus, it can enter into the system. So these are some of the very very interesting things that we have found out by dissecting the genomes of these organisms. And not only that, as I showed you earlier, that this organism is having a very good similarity with another species which is isolated from Antarctica. And of course, we are interested to see whether it has genes responsible for anti-freezing. And to our surprise, we found that it contains anti-freezing genes which are also expressed when we are giving a cold stress. You see the first picture in which this is Rhodotorula and this is the common yeast. And in 30 degrees centigrade, they are all growing together. But when we are reducing the temperature to 5 degrees centigrade, only Rhodotorula grows and yeast doesn't grow. This happens because Rhodotorula has a anti-freezing gene, and which is also expressing in our lab. We have shown that these genes are quite functional. So that means that the genes from this organism can be taken. These genes can be taken, and these genes can be cloned and expressed in the plants which are growing in the cold areas. For example, certain plants don't grow in cold regions. So you can just clone this and you can express this so that the plant becomes resistant to cold bite and survives and produces crops that you are interested in. And Other interesting aspects are phytohormones. So rhodotorola also produces a enzyme which converts. So this is there is a phytohormone called as kinetin. Kinetin is responsible for the cell growth. This is the pathway for cytokinetin synthesis, and uh, this is the active form of kinetin which is taken up by the plant. But the plant doesn't contain this enzyme which converts the inactive form to the active form. And this particular enzyme is present in rhodotorola. So actually rhodotorola helps in completing this pathway. Thus, there is a cell growth and also there is a growth beneficial effect. And actually, Dr. Anindita's lab, they were very interested to see another aspect, that too, they have discovered a very interesting mechanism about this organism. That is, this is not a bilateral relationship. That is, it is not a relationship between higher plant and yeast, but also there is another member in this union is a bacteria. So that study came up after we did the genome analysis. So this is a very beautiful story, and we found this is a nitrogen fixation pathway where you can see that soil takes this nitrite and then nitrate and nitrate reductase and then becomes ammonium, and ammonium is taken up, and the plant or any other higher organism, they produce the amino acids and they are synthesized. And plants directly cannot produce ammonium. Somebody has to provide it. And then when we studied rhodotorola genome, we found that in this process, we know that rhodotorola helps in nitrogen fixation. But then rhodotorola doesn't contain this enzyme. So how is it possible? When this enzyme is not there, so the end product is this. End product is not this. And if the end product is not this, the plant cannot take it. So what is happening exactly? So this led to another set of questions. That is what was taken up in Dr. Anindita's lab. And we went ahead and did further experiments, and we found that there are many there are actually many nitrogen reductase sequences were amplified by the genomic sequence of this organism. Okay. So from this, so then it was actually quite evident. We are thinking that probably rhodotorola contains all these genes. And then when we went ahead and analyzed, we found that these genes are not originated in a higher plant, and that was again done by the similarity search. And this led to the next question is whether there is a bacteria harboring inside rhodotorola. And we went back to our genomic study, and then we found, as I said, that we have done genome assembly, that is joining all the reads that we have sequenced. And after this process, we found that there are some reads, some sequences which are not part of the assembly. Then what we did, we aligned all of the reads that were not mapped to the assembly, which is apart from the existing assembly. And then we analyzed it, and we found that there are about 50 sequences which belongs to a bacteria, Pseudomonas. And Pseudomonas is known to be a nitrogen fixture. And from there, we took our journey to understand whether actually Pseudomonas is residing inside rhodotorola. And Anindita and her collaborators did a lot of experiments and microscopy and TEM and other things to show that, in fact, indeed, that is the truth. You can see the bacteria is growing inside the yeast. The yeast is unicellular sometimes, but when the sexual phase happens inside the plant, it becomes multicellular, and that is when the bacteria is sitting inside the yeast. So this was proved, and this is a very big finding, and this was published in Plant Cell. And this is the hypothesis, actually. This is the discovery we made, that yeast rhodotorola is a single cell structure. It enters into the plant, and it enhances the growth of the plant. And when you take it out, you see that there is a different form. It is dimorphic, it is mycelial. And this bacteria, which is growing outside, then it enters into the mycelium, and then it attains a different form and enters. So this is how the life cycle is completed for this organism. So this story is actually very nice and very inspiring. So as you can see from here, that we could find many interesting genes and enzymes from this organism. And this is an entirely homegrown story in my lab. Diksha Singh, one of my graduate students, she went on a vacation way back in 2013, and she was very curious. In Lakshadweep, here you see all these green spots, and she collected the sample. She thought that what are these growing here, and this nice water. And then she brought it back to the lab and then started. So she's the one who actually isolated and analyzed and found this amazing story about this organism. And this is the extremophile. Why we call it as extremophile? Because it is growing in a sea water. It is halophilic. It is fond of salt. And this is our story. We are expecting, we are thinking that it is having a bacterial association. We still have to prove it. And this also has amazing pH neutralizing ability that I'll be talking about. And it also synthesizes vitamins called as thiamine, and it shows motility that we have seen in our lab. It, although it is filamentous, it doesn't have flagella, but it still moves in a twitching movement. And it is a new species. It is not discovered earlier. It was not identified earlier. This is how it looks like under the microscope. It's a very thin filament, and when it is grown, it develops clumps, and it does not have heterosis. This is cyanobacteria. Cyanobacteria are the ancient blue green algae, and those were responsible for the great oxygenic event. And they do not contain the specialized structure for fixing nitrogen, so they are called as non-heterocystous. And they look like this under fluorescence microscopy. And what we found with these organisms very amazing. We put them in acid, and then immediately we found that the acid is neutralized into alkaline form. And this was repeated, and this is done by these two amazing girls. She is Diksha and she is Shreya. Shreya is a master student in my lab. She's stayed here for a year. So she did most of this pH experiment in different combinations. So we were curious. We wanted to know exactly what happens, whether it is the cell wall which is doing this, or whether it is inside the cell, this is happening, the enzymatic reaction, or what. And then we tested with all kinds of acids. We tested with inorganic acid, we tested with organic acid, and we found that this is more prominent with the inorganic acid, and it happens just within 1 minute. And this is very interesting for us. And then we went ahead to see whether it is happening in the living cell or it is in a dead cell, and it was found to be true in case of living as well as dead cell. Then we took it forward to see whether it is happening in all the cyanobacteria that is growing inside our lab. And this is another interesting story, that this property is existing in most of the halophilic bacteria, halophilic cyanobacteria that is growing in the sea or growing in the saline water. And similarly, we went ahead and did some microscopy, scanning electron micrograph, and then we found that like the arthenium story, it is indeed a surface phenomenon. There is some surface changes are happening, as you can see here, and these are the three-dimensional structures. And we took it forward to see that what is the length, what is the height of the cell wall, or what is the height of the surface that is eroding in treatment of acid. And we found that when the acids, when the material is treated with acid, you see some erosion. That is, the difference is quite substantial. So this indicates that something is happening at the surface level, and we are yet to confirm what elemental changes are taking place. And this we are doing with our sister lab, CGCRI, to see exactly what is happening. And we are suspecting, we are thinking that maybe calcium carbonate is responsible for this kind of interaction. And we went ahead and did similar experiment, and we found that calcium carbonate indeed does similar changes. But also there is a change, that is, there is a difference. It also works with the organic acid, which is not happening in case of our organism. And we wanted to again study at the cellular level, at the gene level, and then we sequenced it. And the number of scaffolds generated, that is number of fragments of the genome that we generated, the lesser the better. And we have got about 16, which is a good number. And we have also found the genome is complete to 98% level. So it is almost complete. And this can be used further for understanding the genes it is encoding and what exactly happens inside the cell. And we did a phylogenetic analysis. Phylogenetic analysis is something where you have the entire genome, and then you compare it with the existing. NCBI, as I said, is a repository which every time somebody sequences, they have to deposit it there. So this contains the universes, all the sequences that are sequenced in the universe, and then we compare them together. And this tells us what is the tentative species name, and it is closer to Halomicronema, which is, but then it is only 92% similar. That means it is eligible to be called as a new species, which is not named as yet, since we have not published it. And we also did multiple analysis, finding how many genes are unique in this, and then we found 4 other species which are closer to that. And this contains 16 new genes, which is not 32 new genes, which is not present anywhere else. So those are the new genes, and those are very, very interesting genes, and we are looking at those right now. I'm not getting into details of the mechanism. And we are also, as a lab, we are interested in also production of biofuels and fatty acids, and we want to give stress so that we can see whether the fatty acid composition is increased. And we found that when they are under stress, that is when they are in acidic stress, the composition, the fatty acid production of fatty acid is very high. So this is something also in our radar. We are trying to understand, analyze this. And Diksha did also something called as transcriptional profiling. What is a transcriptional profiling? Genome, as I talked about, genome is sequencing of the DNA. Also, there is another technique where you are taking the entire RNA sequence of the organism and you are rapidly sequencing. This tells you how many mRNA were transcribed from the genome. So this tells you how many genes are actually actively transcribed. And this is a technology largely used for finding the genes responsible for a certain phenomenon. And we did a 6-way comparison between the pH stress and nutritional stress, and found that between the nutritional stress, we have large number of genes are perturbed. And this is under study. And this is a different plot to show the differential. This is a different plot. This is called as a volcano plot. And this tells us the different number of genes differentially regulated are high between C and D. This is under study. And we are interested to understand what are the genes responsible for this kind of change, or whether we can take some of the genes and then clone it in a plant or some other species to make it acid resistant, or use it for bioremediation, or whether we can use just the biomaterial as in case of Arthrium malaceolum. So this is undergoing, this is continuing, and we have not concluded this story, but in very soon we would like to develop a biomaterial from the cell wall, and we would like to decipher the exact mechanism that is happening on the cell wall so that we can use it for bioremediation. So with this, I would like to thank actually the people who did all the work. I'm just presenting their work. And this is Dr. Anindita Sil from Calcutta University, and this is a student. And this is Dr. Rajiv Majumdar, who did all the arthrium work. This is Dr. Diya Sen, who worked with Rhodotorula. And this is Diksha, and this is Shreya, who worked on our PK species. And funding agency CSIR and DPT, Department of Biotechnology, and ICR is greatly acknowledged. And this is my lab family who actually are doing everything. I'm just talking. So thanks a lot for giving me this slot for presenting our work.
Thank you, Dr. Sucheta, for such very informative talk today, giving a glimpse on how the naturally existing microbes you have utilized it for various sustainable application, be it environmental application or any other application. So I would request some students to put their questions here, and some questions are already there, so I'll take those questions.
Sure.
Uh, the question that comes, let me read it. Uh, One of the students is asking how the parasites switch to endophyte. Is this only for the nitrogen fixation gene?
Okay. So this is actually a very long question. This is a very good question, but it will need a very long answer. That is, switching between parasitism to endophytism doesn't happen in a day. It happens over a long period of time, and the parasites actually adapt depending on the environmental condition. And when the parasite becomes an endophyte, it establishes connection, or it establishes connection with the host. It enters into the host, evading the host defense mechanism. But once inside the host, it has certain pathways, or it has certain genes which in turn helps the host, and the host also provides some of the nutrients. So that is how they develop a mutualistic relationship, and that is how they develop together, they grow together. And along the side, if they are finding a third organism, say bacteria, and if the bacteria is existing, like along their journey they found another organism which can be very beneficial to them, then they take it. And during natural selection, if that is useful for all of them, then it stays on. If it is not very useful, then it is dropped by the nature. That is how it happens in reality, over a period of say thousands of years.
Okay. So next question I'm taking from another platform. There's a question Shri asked Shri Hari. Can hoax gene editing done in human? Can it be done in human?
Gene editing, yes, it has been started, but there are ethical issues. But gene editing experiments are done in humans already. Many different labs are doing. There are CRISPR Cas technologies, and there are many other technologies for editing the genes. And this is actually in practice. But then, in humans, it has a lot of ethical issues. You cannot change a gene, and you cannot make that person available, or put all the evil genes or good genes together, and then make a human or a synthetic human being. Uh, Although it is technically possible, but there are guidelines to work on those.
So next question again from Shri Hari is regarding moisture condition. Are moisture condition also factored for the selection?
Which conditions?
Moisture condition. I'm not sure the question asked by the student is correct or not.
Moisture.
Yeah.
Yeah, I'm not very sure if I understood the question. If it is if the question is about whether moisture is required for establishing connection, that is again, water is required
Yeah.
for everything.
I
So
will
that,
request the viewer Manoj Patnaik to frame the question again if this question is
okay.
not clear. And then there is interesting question again from Shri Hari. Can the gene editing in mosquito be done so that it does not spread disease?
Yes, I think this is a very interesting proposition. In fact, it is already in existence. Like in mosquitoes, there are genetically modified mosquitoes are there, which are not biting humans, and those mosquitoes are fighter mosquitoes. They can grow very fast, and they can also kill the mosquitoes which are spreading disease. So that has already been tested, and I think Bill and Melinda Gates Foundation has sponsored a project, and those mosquitoes, transgenic mosquitoes, were released sometimes back.
Okay. So one last question we will take since we are reaching to the time limit. The question is, in the endangered species like tiger, is it under nutritional stress. So, what is the major reason for like decrease in their number?
Yeah, in natural selection, nature eliminates those which cannot survive in the existing condition. So it could be anything, maybe temperature, it could be food, it could be any kind of stress. Like earlier days, there are many organisms were existing. For example, several types of birds. They are not existing now because rapid urbanization. They are not getting space. Similarly, spiders and other organisms, if their natural habitat is to grow in a certain place and have a certain type of food, which probably when it is not available, they are eliminated with the existence of existential stress in fact.
One of the student, probably the teacher also asked about your paper, if you can share about the pseudomonas inside the yeast.
Okay, so my paper had my presentation had the reference to the paper, but if you want, I can share it. That is a plant cell paper, so they can just search my name in PubMed in 2020, and that plant cell paper contains the whole thing.
Okay, so with this, thank you very much, Dr. Sucheta, for this very informative session. And it's really a lot of information. Students might have gained new information how even they can explore natural resources. Like I can see your students, even in their holidays, they could explore the new things and demonstrate it for very good application. So thank you very much for this session, and I also thank the students for staying with us and putting up good setup of questions. With this, I thank everyone. We shall meet again in next webinar under this series. And last not the least, I must thank the team at CSR IICB who is always enthusiastic to have very good lectures under this JIGYASA-ATL webinar series and even coordinating this lecture. So thank you everyone. Take care.
Thank you
Goodbye.
so much.
Happy Diwali in advance.